some students represented that they have been already connected, and after a gap of 3 or 4 days, I am coming in my live class. Actually, I was engaged in some other work and some household and some research related work. And today we are going to discuss fundamentals of the genetic polymorphism. I am just saying that we are discussing fundamentals of genetic polymorphism because the genetic polymorphism is a most important topic nowadays for the research and study, and it is included in the syllabus of postgraduate as well as in the NET examinations. So, at first we understand that what is genetic polymorphism, and it is very simple, and we simply say that the genetic polymorphism refers to the occurrence of multiple forms of total population is represented the gene pool, and in the gene pool some variants of genes are present on a particular locus, and that variants or various forms of the gene present on any particular locus is simply referred as the genetic polymorphism. And these variations, of course, if it is the gene, then the gene is present in the DNA, and the variations are encoded in the DNA sequences, because the site where DNA gene is present is DNA, and gene is nothing but the smallest stretch of deoxyribonucleotides. So the variants of the sequences present on the particular locus is responsible for the formation of genetic polymorphism, and the genetic polymorphism is determined by the differences in nucleotide bases or change in the structure and function of the gene. So, the genetic polymorphism is actually represented the different forms of the gene present at particular locus. And a very simple example we consider that the ABO blood group. And if you consider the ABO blood group system, the gene is represented for the synthesis of antigen responsible for the blood group A, or the gene responsible for the synthesis of antigen B on the B cells blood group, whereas in the O there is no antigen like A and B, but it does not mean that O has no antigen. A has also antigen, O has also antigen referred as the H antigen, and that H antigen is responsible for the synthesis of, provide the base for the synthesis of antigen A and antigen B. And a fourth one is reported where the H antigen, the gene responsible for the H antigen, is referred as the FUT1 and in the condition of the FUT1 mutation, it is not synthesized. Now the H antigen is absent in the O. So here the ABO blood group, the gene which is responsible for the synthesis of ABO blood group is responsible for the polymorphic nature. But one thing more, which is necessary to understand, that the H is synthesized by the another chromosome, and AB is synthesized by the another chromosome. For example, the ninth chromosome is responsible for the synthesis of blood group antigen for the blood group A and B, whereas the H antigen is synthesized by the chromosome 19. So here the H does not represent the polymorphic nature, because the position of the position for the gene which is responsible for the synthesis of H antigen is on the 19th chromosome, and the gene which is responsible for the synthesis of A or B antigen, particularly the enzyme, the gene which is responsible for the synthesis of the enzyme transferase, and that specific transferase is responsible for the synthesis of antigen A and B. So, in the conditions of the ABO blood group, if we exclude the H antigen, then the ABO blood group represents the three genic component. One, which is not responsible for the synthesis of the particular type of the enzyme transferase. Second, which is responsible for the synthesis of the enzyme transferase for the A, and the third, which is responsible for the synthesis of the enzyme transferase for the antigen B. So here we observe the polymorphic nature of a gene. One gene represents three variants. So the condition is referred as the polymorphism, genetic polymorphism. Likely, when we study, just I am saying that today my class is confined to the fundamentals of the genetic polymorphism. I am not going to explain the genetic polymorphism in detail. So I confined my speech for the fundamentals of the genetic polymorphism. And here, the second example which represents the genetic polymorphism is the human leukocyte antigens. And in short, it is referred as the HLA system. And when we use the word system, that means the HLA or human leukocyte antigen is more than one in number. That means it represents the variants, and the HLA system is a highly polymorphic group of genes involved in the immune response, and variations in the HLA and genes influence an individual susceptibility to certain autoimmune disease or transplantation, compatibility, and the defense against the infectious agent. So here the HLA leukocytic antigen, human leukocytic antigen or HLA represents the genetic polymorphism. When we study, there are a number of the examples represented the genetic polymorphism. And it is very simple to understand that the genetic polymorphism is responsible for the genetic polymorphism is defined as the gene present in any population in various forms. That means the gene or the population represents genetic variability or genetic polymorphism. Polymorphism more forms of a particular component. That means the polymorphism. So if the gene represents the variants in any population or in any gene pool, then it is considered as the genetic polymorphism. Second point, which is necessary to understand, that what are the causes of genetic polymorphism. The genetic polymorphism can arise from various mechanisms. It is not one particular mechanism which is responsible for the development of genetic polymorphism, but the genetic polymorphism is developed by the number of mechanisms, and some are, if you consider, like the mutation. If you consider the mutation, that genetic mutation such as point mutation, insertion of the any small sequences of the nucleotide, deletion of the any small sequences of the nucleotide, and chromosomal rearrangements, all are the functional source of genetic polymorphism. Because when these components or when such type of mutations are occurred in any particular gene or functional gene, they represent some variation in their expressiveness, and in this way they are responsible for the development of genetic polymorphism. The mutations can occur either spontaneously or due to environmental factors. Either the mutation is caused spontaneously, or due to the environmental factors, or chemical expositions or exposures, or errors during the DNA replication. All such components have the all such events have the mutational impact, and they are responsible for the development of genetic polymorphism. The second cause, which is responsible for the development of genetic polymorphism, is genetic drift. The genetic drift is also responsible for the development of genetic polymorphism. And here, the genetic drift represents random changes in allele frequency within the population, can lead to the emergence and maintenance of genetic polymorphism. The genetic drift is particularly significant in isolated population or those with limited gene flow. And the genetic drift in a nutshell, I was described in my offline class. And in that class, I represented schematically that some populations were migrated in the to the another new geographical area, and in that condition they are responsible for the development of a new allelic frequency or new gene frequency, establishment of the new gene frequency, and that the new gene frequency may causes the genetic polymorphism. The third important cause is the natural selection. And we all know very well that the environmental factors and selective pressure can favor certain alleles over others to increase the survivalship fitness, and in this way they influencing the prevalence of genetic polymorphism. Now, for instance, alleles that confer advantages in adapting to a specific environment or provide to disease may become more common in a population over time. So, here the natural selection is also responsible, is also a important cause of genetic polymorphism. Now, the next cause of the genetic polymorphism is gene flow. Now, the gene flow occurs when I also discussed the gene flow in my line class, and the gene flow, if we consider the gene flow occurs when genetic material is exchanged between different populations through migration and interbreeding. In my class, I schematically represented that some people of any population married to other people, individuals of the other population, and come and live in the parental stock. So some new genes are included in the parental population, and that the parental population may represent the genetic polymorphism. Hence, the gene flow is also a cause for the development of genetic polymorphism. Now, the non-random mating, the another factor, the another phenomenon or other event which is responsible for the development of genetic polymorphism is the non-random mating. And the non-random mating, I also discussed in my offline class. In non-random mating, the individuals of any population is selective breeder. And when the individuals are selective breeder, they prevented the random mating or random interbreeding of individuals in any population. And certain mating pattern in this condition, in this type of mating pattern, the such as the assorting mating, that means assorting mating means the mating by selection, or the choosing partners with similar traits, and or desortive mating. The assortive mating means the choosing of partner with similar traits, or desortative mating, that is the mating among mating between the partners with dissimilar traits, and that nature of the mating or non-random mating is responsible for the development of, or can lead to a trend for the development of genetic polymorphism. In this way, the genetic polymorphism is caused by a number of events, or ecological, or evolutionary or genetic events, and the variants of the gene is developed in any population. Now, the third point is the types of genetic polymorphism. What are the types of genetic polymorphism? And here, on the basis of the nature of nucleotidic segments or sequences, or the number of nucleotides involved in the development of genetic polymorphism, the genetic polymorphism can be differentiated into different categories. First one, or the simplest one, is the single nucleotide polymorphism. The single nucleotide polymorphism is also referred as SNP. S for single, N for nucleotide, P for polymorphism. Single nucleotide polymorphism. And the best example for the single nucleotide polymorphism we all studied, that is the sickle cell anemia. And when a single nucleotide at the sixth position is mutated, then the glutamic acid is converted into the valine, and that is responsible for the sickle cell anemia. So here the only one change in the only one nucleotide is responsible for the changing of the nature or nature of a trait. That means the nature of the beta hemoglobin is changed as sickle cell stage. So it is a best example of the SNP. And not only the sickle cell anemia, but SNP are the most common type of genetic polymorphism. And as I say, that they are characterized by a single base pair change in the DNA sequence. And when the single nucleotide base pair is changed in a gene or in DNA sequence, they occur at specific positions in the genome and, and can influence gene expression. And likely, as we observed in the sickle cell anemia. And the gene expression, if the gene expression is changed, it is responsible for the changing of protein structure and its function. So here the one nucleotide, the change in one nucleotide is responsible for the change in the configuration and function of any protein. So it is the most important polymorphism observed among the population. That is the single nucleotide polymorphism. Second one is the indel sequences. Indel is formed by two words. One is in represents the insertion and del represents the deletion. That means some nucleotidic sequences belongs to indel category, and indel refers to the insertion or deletion of nucleotide bases in the DNA sequence. They are unstable, and these insertion or these variations can affect gene functions by altering the reading frame or distribution of the nucleotidic sequences. And suppose when the some sequences are inserted, that means it is responsible for the changing of the reading of the frame of the nucleotide in the term of triplet, or if some are deleted, then it is also responsible for the changing in the reading of the sequences. So the shifting of the reading in the condition of the insertion and deletion of the small sequences is affected the expressiveness of the gene, or the nature of the protein synthesized from the gene, or it may causes the various genetic disorders also. So the indel is also an important types of genetic polymorphism. Third one is the copy number variation. That means copy number variations are genomic rearrangements that involve the duplication or deletion of larger DNA segments. In some genes, when we study the genetic polymorphism, we observe that in some genes, the larger segment of the DNA either duplicated or deleted. That duplication and deletion of a particular roughly moderate number of the nucleotide is responsible for the genetic polymorphism, and that stretch of sequence which is deleted or duplicated is referred as copy number, and that the number of the copy present in the present at any locus of the chromosome is responsible for the variation in the gene, and now it provides also a type of the genetic polymorphism. Now, the genetic polymorphism third type is chromosomal rearrangements. Sometimes the chromosomal rearrangements involve large scale structural alteration in chromosomes, such as inversion. We all studied the chromosomal structures are changed due to the inversion, translocation, or duplication. That the inversion, translocation, and duplication, these rearrangements can have adverse effect or have the diverse effect on gene expression, and that the gene expression is responsible for the development of genetic polymorphism. And we observe that a number of genetic diseases or genetic events are associated with the such events are associated with the individual health and evolution. So these are the types of genetic polymorphism. Now we understand the importance of genetic polymorphism. What is the importance of genetic polymorphism? The genetic, just we discussed that what is the cause of the genetic polymorphism, what types of the genetic polymorphism, and what is the genetic polymorphism. Now we understand what is the importance of genetic polymorphism. Genetic polymorphism, if we consider, then the genetic polymorphism play an important role in various facets of biology and genetics. It is not. It has no. It has no single importance, but the genetic polymorphism has a number of importances in the biological system, and some important importances are as the evolutionary adaptation, disease susceptibility, biodiversity conservation, forensic identification, agriculture and livestock improvement. So the genetic polymorphism are important in a number of events or in number of processes. First, if we consider the evolutionary adaptation, then the genetic polymorphism is the driving force behind evolution. Because when the genes are not polymorph, then there is no chance of the selection or rejection of any gene from the population or for the population. बिकॉज सपोज कि आपके पास एक ही रोटी है और आपको बोला जा रहा है कि आप रोटी खाइए, देन यू हैव नो अल्टरनेट, यू हैव नो ऑप्शन टू सेलेक्ट एनीथिंग बिकॉज you have only roti, but you have option that you select either roti or puri or naan. Then you will have you will have option to select anything. So the genetic polymorphism provide the conditions for the evolution and provide raw for the evolution on which the evolutionary events can occur or can operate. Now the evolution in this condition allowing population to adapt to changing environment and pressures. So in this condition, if there are a number of variants are available in any population, then the population has option to select anyone or to evolve or to protect from the environmental pressure or to modify as in changed environment. So the genetic polymorphism provides the genetic a basis for the evolution. Now if you consider in nutshell, it provides a diverse pool of genetic variations. What I am saying, it provides a pool of the genetic variation from which advantageous traits can emerge, improving survival and reproductive success. Because in classroom, I told you that the main point for evolution main points of the evolution are to increase the rate of survivalship or to increase the reproductive fitness. These two are the major points. Of course, some minor points are also responsible, but these two major points are responsible for the evolution. So it is the first importance. The second importance, that the genetic polymorphism provides disease susceptibility and drug responses. Disease susceptibility means the variation in genes due to genetic polymorphism can influence an individual's susceptibility to certain disease, or their response to medications, because the gene is responsible for the all physiological activities, directly or indirectly. And so there are, if the number of variants are available, so it provides the susceptibility or provides a condition to response in the case of medication. Understanding these genetic variations can, in personalized medicine, enabling the better outcomes of treatment. So here, the genetic polymorphism is also responsible for significant for disease susceptibility, and drug uh, responses. The third one is the biodiversity conservation. It is the it is another importance of the genetic polymorphism to increase the biodiversity conservation, because the genetic polymorphism contributes to the biodiversity of a species, providing the necessary genetic variation for population to adapt and withstand environmental changes. So it provides a condition for the existence of more diversified populations or more diversified individuals of any population. In this way, it provides help in the biodiversity conservation. Then the polymorphism, when conserving the biodiversity, then it preserving the genetic diversity that is crucial for the long-term survival and conservation of a species. It is the important. The next importance, or the fourth importance, is to develop or is to improve agriculture and livestock for the economic development. Here, we develop the more and more variants of the gene, and then, of course, it is the technological program and used in the applied part of the life sciences, but it is more important because more and more diversified components are the if the genetic polymorphism is observed, it plays a vital role in breeding programs of animals, enabling the selection of desirable traits of crop and livestock. So it provides, if you observe the genetic polymorphism, then they have the different natures, different functions. So the different nature and different functions we provide opportunity for the selection of a particular gene to improve the agriculture products and livestock. Now, the third one, fifth one is also applied. That genetic polymorphism is used in the forensic identification. It is the forensic identification and the polymorphic DNA markers. When they are the polymorphic, then it develops the marker, a specific marker for the particular person or particular individual. And then the polymorphic DNA markers such as STR, and the full form of the STR is short tendon repeats. And probably if you was studied the DNA fingerprinting, the term was also available there. That STR, the small tendon repeats, or VNTR, very small tendon repeats. All are the all provides the marker and widely used in the forensic science, as in the DNA profiling and identification purpose. So the unique genetic variations observed in different individuals, and that unique genetic identification help establish, identify, and aid in solving criminal offenses and criminal cases, or of course in the parental claims. So The genetic polymorphism or very small number of tendon repeats or tendon repeats or a small tendon repeats, all are responsible or are examples of the genetic polymorphism because the same sequences are repeated and again repeated in any genomic sequence. Now, the uh, next component, if you want to study here, the what is the impact of genetic polymorphism. Just when I initiated my today lecture, I told you that today we are just confined in fundamentals of the genetic polymorphism, and from the next classes we will discuss more in the parts or the specific topics related with the genetic polymorphism. So I will take 3 or 4 classes on the genetic polymorphism because it is important both for your postgraduate examinations and the NET examinations. And the some questions are always asked in your GRF NET examination. So now I am representing the impact of genetic polymorphism. What is the impact of genetic polymorphism? And the first one is the genetic polymorphism can impact traits and disease susceptibility. It is the first. Second, certain single nucleotide polymorphisms are associated with increased cancer risk, including breast cancer and colon cancer. And there are a number of research is initiated on this topic to identify the polymorphic genes associated with the different types of cancer. Next one is the copy number variants. And one important, that is copy number variant, just I discussed. The copy number variants, that is, for example, the deletion in CYP2D6, it is a sequence. Copy number variants I am discussing. The copy number variants, an example is the deletion in CYP2D6 affect drug metabolism. It observes the deletion, it represents the affect the drug metabolism, while duplication in AMY1 influences starch digestion. So here the copy number variance is also effect on the metabolic activities. Now third one, that is indel. I am just giving the examples which is associated with the human welfare. And indel of the allogene, ALU1, ALU1, that is the endonuclease enzyme, and that a number of researches occur throughout the world on the indel, ALU-indel sequences to study the evolutionary pattern and genealogy and diversification of the human population throughout the world. Now second, the indel gene like PCSK9, a reduced heart disease risk. So the indel or the gene developed due to the indel has also significant for the study of or prevention of the disease. Now, the next, a small repetitive area and referred as the microsatellites. If you are not understanding properly the microsatellite, then you consider a small sequence which is repeated and present at a small area. Repetitive, a small area. That is referred in present at any chromosome, and that is referred as the microsatellites. And the microsatellites provide genetic diversity insights and are used in forensic, paternity, and conservation genetics. Third one or next one is the mitochondrial DNA polymorphism. And we all know that mitochondria is inherited from mother only. So the mitochondria present in a population throughout the globe is inherited from maternal stock, and the mitochondrial DNA polymorphism affect mitochondrial activity linking to disease like diabetes and Parkinson's. So it is also the useful for the study, that is the impact of the genetic polymorphism. Now the HLA polymorphism, that human leukocyte polymorphism, influences immune response. Just I told you, it influences the immune response and disease vulnerability with a specific a specific variant linked to autoimmune disorders. So the number of autoimmune disorders is studied and present in the human being, which is linked with or which is associated with the HLA polymorphism. Now, the HLA polymorphism is also impact on organ transplantation reactions due to their role in transplant rejections. So these are the actions or importance of the genetic polymorphism. Now it is necessary to know what is the applications of genetic polymorphism. And the first and important application of the genetic polymorphism is the genetic polymorphism finds application in medicine, just I am saying, forensic, and non-conservation genetics, because it is changed, it represents the variant. So the gene is non-conserved here. Second, in personalized medicine, it predicts individual responses in medications, including adding in the treatment plan. Third, SNP, that means the single nucleotide polymorphism, can anticipate responses to chemotherapy and assess the risk of adverse drug reactions. Now, the next, it is the applied part, and sometimes asking in your NET examinations that the genetic polymorphism information or collection of the information from the genetic polymorphism informs conservation strategy for the long-term survival of endangered species. Next, just I am saying that allogene is responsible for the ancestry. So, the ancestry testing utilizes mitochondrial DNA polymorphism and Y chromosomal polymorphism. And that DNA polymorphism to trace maternal and paternal ancestry, because the mitochondria is always inherited from maternal stock, and Y chromosome is always inherited from paternal stock. So when we want to study the ancestry either from the maternal lineage or paternal lineage, then we study the polymorphic natures of the mitochondrial genetic material and the genetic material present in the Y chromosome. Now, the genetic polymorphism, I am just saying, in agriculture or in the livelihood, we selected the desirous genetic material to improve the crop and livelihood. Now, second, if we identify the SNP for a particular associated disease, then we develop the resistance, or we analyze the nature of disease, or we will able to work on the gene therapy by only correcting a single nucleotide base. So the genetic polymorphism has a number of applications. So in this class, I just give you a brief outlines of different aspects of the genetic polymorphism. What is genetic polymorphism? What is important? Which events is responsible for the development of genetic polymorphism? What is the significance of genetic polymorphism, and what is the application of the genetic polymorphism? Now, I give you the time, 1 or 2 minutes for your question, and if you feel problem in any part or any concept, then you write, you give me message, and then I will either explain you through message or give you reply in next class. Have you anyone has any problem? Then you give me message. If you have no problem, give me no problem. Then I am going to terminate my life. Sunil is writing that low polymorphism, that is low adaptivity or extinction risks. Of course, Sunil, just I am saying that low polymorphism is, of course, it provides the conditions for the low adaptivity, because the variance of the gene is not available in the gene pool. But it is not certain that it provides the extinction risks, because if suppose if 1 or 2 variants are present and both are selected in the population with respect to the particular environment, then there is no chance of extinction risks. Of course, if we consider the component in term of the unstable environmental conditions, then your representation is correct. Sunil, do you understand properly? Sunil, do you understand properly? Give me message. Because your representation is correct in the conditions of the unstable or early environment, but it is not correct for the established population or population present in genetic equilibrium. So the reply is depends if the question is asked in your examinations net or JRF. So then it will depends on the body of the questions that the question what will be asked by the body. Now, as I suppose that no one has any question, so I am very much thankful, and due to some important work, always also some personal work and some research work, I was not coordinated you through online classes, but I will try to present in the regular contact through the live classes. Okay, thank you to all.